and all of the transcribed pieces of RNA in the cell are the transcriptome of that cell. So those are the actual things that are expressed in that cell that go on to become proteins that do the work in the cell. So that's what we're talking about. And metagenomics, if we throw a meta in front of any of these terms, we're just talking about, instead of sequencing and reading the DNA from or RNA from a single cell, or maybe from a single organism, we're grouping together the DNA, the sequence data from a bunch of different organisms, maybe because we can't separate them, like we're sampling from an environment in the case of metagenomics. We don't know what's there, we just want to find out. We get all the nucleic acid information from that environment, and it's all mixed up amongst all these different species. So that's metagenomics or metatranscriptomics. All right, and then this is the graph we have to show with every genomics talk, where we're talking about sort of big data applications and the very rapid decline in the cost of sequencing, which means that you can produce much more sequencing data than was ever possible before, going from when the human genome was first sequenced, taking 10 years and costing something like $3 billion. And now we're down to the $1,000 genome that you can sequence, I don't know, in a day or less. I mean, I can't remember what the exact time frame is now, but very, very quickly, depending on what technology you're using. So a lot of scientists out there, a lot of biologists, those who are computationally inclined, those who are less computationally inclined, want to use these data to study the fundamental understanding of how things work, how biology works. And so this is a substantial growing need for help in this area. And there are various resources available, of course, ECSSD. There's another SF project that used to be called iPlant, which is now called Cyverse. I work together with a group that's called the National Center for Genome Analysis Support. Originally started in Indiana, and PSC is now a partner with that. There's web platforms out there to help people do these things. One well-known instances of that include Galaxy and Gene Pattern, and many other resources available. So as we work with these communities, I find sort of some key challenges that make up most of what people are having trouble with, although I've left a few of them out of here. But some of these are just wrangling the data, just managing it, getting it where it needs to be, getting it through the pipelines and the analyses they want to run. Actually choosing what software should be used and knowing how to appropriately apply the available tools to get good results is something that's difficult, especially because the landscape for software is constantly changing, in part because the data landscape is also changing fairly rapidly with new technologies for actually reading the data, sequencing data, and different characteristics of the data, different error profiles, different lengths of the reads that come off the sequencing machines, and these kind of things. So the tools are constantly changing, and people are constantly developing new algorithms to deal with these. So it's kind of a headache and nightmare for everyday scientists, especially if they're not focused completely on bioinformatics to manage this. And then when they find the tools, or if they find the right tools, a lot of these have very large memory or other computational requirements depending on the algorithms used and the size of the data people want to deal with. And putting all these different tools together in some sort of a pipeline hits issues of reproducibility and correctness, and how do we work on that front to make sure that other people can reproduce what we're doing. So these are common issues in this arena. So just to illustrate this is a smattering of genomic software that we've installed just on bridges over the last few months. We've been getting as we've been getting it up into production and running. And these are, this is, we have many outstanding requests, still to go to, and there's many others that we've had on other platforms that we haven't yet gotten on bridges. But there's a lot of stuff constantly being developed, and so just installing and maintaining these tools is one of the one of the biggest things that people come to us for. They don't want to deal with this headache of having to deal with all this, and so this is something that's big on people's list in terms of biologists, of why they love to come and use Exceed, because we're dealing with the software, we're managing it, and helping them that way. So I've been involved throughout my time at PSC with Terragrid and with Exceed, and through now in Exceed with the Extended Collaborative Support Service. And as I've gotten into this genomics realm, it's opened up a little bit different flavor of ECSS support. The sort of the canonical, the standard ECSS project is that you get with one research team and you go in depth with them on some problem, and then you maybe highly optimize a specific code, and this is the main code that they use for the research, and then they go on, they're happy because this thing that is their main workhorse now runs much better. And in genomics, because of what we just mentioned, just what I just mentioned about the constantly changing nature of the landscape, the many different tools, and how things are done, there's just too many tools except for a few. There's some that are very heavily used by everyone, but there's a lot that are just sort of on the fringe, or that are some people use one, some people use another, and it changes frequently. So we don't generally folks aren't generally interested in us taking one code and spending a lot of time and optimizing that code. They want us to optimize their workflow, and they want to find reasonable tools for the job and have it perform reasonably, and get the work done, and then they move on, and then in 6 months they'll be using a different tool, and so this is how it goes. So they generally want to know, what tools should I use right now? Where can I actually run these? Because sometimes they have requirements that I can't meet on my local resources, and what national resources are best suited for them? How do I actually run them on those resources, and how do I get allocations of time, in particular on Exceed, to do this? So in my ECSS experience with genomics, I'm generally, and this is sort of setting the stage for what I'm going to the projects I'm going to be going through here, and why I'm talking about 3 of them, or maybe 4 today instead of just 1, is that I'm primarily allocated through this novel and innovative projects arm of ECSS, whose mission is to reach out to new communities, new users, those that are not real familiar with using these high-performance computing national resources. And so in fact, all my time right now is allocated in that sort of an arena where we're rather than doing real in-depth projects with one research group over a long period of time, we're spread out amongst many different people who need this kind of support that I mentioned here, answers to these questions that I've put up here to help get them moving along. And so there's a combination here of what we'll be talking about today, of sort of this novel innovative projects, reaching out to various different groups, and also what I'm calling light ESRT projects, which are the traditional ECSS projects of working with a single research group. Some people have a little bit more need for intervention, for help, and so we create a formal project with them and move and do a little bit more with those groups. But even in these cases, we're again sort of going through just helping them get these pipelines running and working in a reasonable way on the system. So without further ado, let's see. Oh, one more point I wanted to make. We talked sometime in Exceed, one of the big missions is helping new communities and getting new communities on our systems. And so there is some question has been raised, is bioinformatics actually still a new community? They're actually using these systems a fair amount, although I think their overall percentage is still pretty low, maybe like 5% of usage, or I think is the number that I heard at some point, although don't quote me on that, but I think that someone did that analysis at some point. I couldn't find that number, though, to refer to, but something along those lines. It's not a huge percentage of the total usage of Exceed, but we do get quite a lot of requests, quite a number of people. So just to put this in perspective, though, this is the graph of active users on Exceed over the years, up to 2016, where we've got between 9,000 and 10,000 users. And so then putting that in perspective, if we look at the number of registered users on the Galaxy main platform, this is this tool, this web platform for running bioinformatics genomics analyses that was established, I guess, back in 2006. And so that, I don't know if you can see the little graph that shrunk. That's about the scale in terms of about 10,000 users on Exceed. We've got about 90,000 registered users on Galaxy Main. So my point is that there's this huge contingent of less savvy people who want to do genomics, in terms of less computationally savvy folks who want to do genomics, biologists and others, and a huge demand for these kind of gateways that abstract some of the difficulty in doing that and creating workflows and running them on hardware. So, so that's just setting the stage there. My focus has been a lot on the genome assembly problem, because this is traditionally some of the most intensive, it's one of the most intensive applications that we run into with genomics, and because we're reconstructing a genome that has millions and millions of nucleotide base pairs, it has very repetitive sequences throughout these billions of base pairs, but we're generally trying to do this with fragments, reading fragments that are only a few hundred base pairs long, and constructing that back into the full genome is very difficult, especially because you have systematic and random errors to deal with. So you need deep coverage. That means big data, a lot of data, and oftentimes the algorithms do this on in a shared memory threaded parallelism type of a framework, although I'll show one exception to that today. And this is just, again, a word I'm going to throw out there at some point in this talk just to be on the same page. When you're looking at the quality of these genome assemblies, one of the main things that people look at just as an initial sort of sanity check on how well you did is how big are the contiguous sequences, or contigs, that were assembled. And the N50 size is the statistics that is used for that, and that means that whatever that number is, 50% of your genome is in contiguous sequences that are at least as large as that N50 value. So that's just for future reference. So this first project that I'll jump into here is a project that doing the analysis of metagenomic populations with Pingma University of Georgia. And so again, we already talked about metagenomics, where you're sampling from the environment and then assembling all the data together. And in this case, they're looking at all types of environments, where they're starting with human microbiome data, so diabetic patients and patients with inflammatory bowel disease, looking at the populations of the metagenomes in those patients, and looking at how they differ from healthy patients, and if this might be a cause of some of these diseases. And so they're developing a statistical method that can distinguish between closely related species, even if those species haven't been previously characterized. And so they take a huge sample, and they assemble it, and then they do some analysis to bin these different species, and to isolate the different species based on those assemblies. So they just had no idea where to start, though, with these huge metagenomic data sets. It takes a lot of sampling, like I said, of data, these big data sets, and they want to pool them across patients to get better results. And so for the assembly, we were just able to guide them towards an assembly. It's actually MPI based. It's called Ray. It does a really good job with these metagenomic data, and because it's MPI, you can scale it up to pretty much any size of data that they want to do. And so we're able to get them to assemble these metagenomic data sets and guide them on how to use the Ray assembler effectively on bridges, what core counts to use, what data sizes and such. And then for their downstream analysis, they're developing this R-based statistical tool they call Metagen, and we help them parallelize those R-based statistical analyses and distribute their jobs, these data parallel jobs that they have across many bridges nodes, in this case just using capabilities within the Slurm jobs management system that we're using on bridges. So so they've had great results from this, and my time is short, so I'm not going into a lot of detail on any of these, but hopefully just illustrating sort of some of the types of projects and types of issues that people are running into. But they were able, as I mentioned, assemble these data from these normal and diseased patients, characterizing how many different species. The first estimate, sort of what they think is an accurate estimate of the total number of microbial species in the human gut, which around 2,000, and then identifying important species that may contribute to pathogenicity in these patients. So this is ongoing work, but they've had good success and been very happy. And again, a lot of this was just facilitating the data wrangling and the optimization of how they were able to run these tools on and match the tools to the system and this sort of thing. So moving on to the next one here, time is short. We worked on this project with a group at Texas A&M, and this is in fact a we. Raminder Singh from Indiana actually was the person doing most of the hands-on work with this group. And as I mentioned, I've been mostly, like I said, spread out amongst a number of groups, and I was sort of giving guidance on this project, but Raminder was the one who did most of the hard work here. But this was another case of where they're looking at this transcriptome data, and the question is, how do we know, how do we know if how high quality these transcriptome analyses are, and how can we improve the quality? How can we analyze the quality of these things? I'm going to skip that one. And so what they did was they took this highly characterized sort of standardized data set, a standard RNA-seq data set from these well-studied human samples. And these data were generated at 6 different sites across the United States. It was coordinated by the U.S. Food and Drug Administration. And so they had a couple of different samples, 10 pooled cancer cell lines, and then another sample that consisted of multiple brain regions from various donors. And so the idea here, using these well-characterized data sets, is that they can take these data that have been well studied, run them through a pipeline, and look at different aspects of how you might change the pipeline, or how you might also look at the results across these different sites that generated the data, and start to pin down what are some factors that influence the quality of the result that you get when you assemble and analyze these transcriptome data. Okay, so I think I just said everything on this slide. So they're exploring the factors that influence the quality of the assembly and subsequent analysis of the transcriptome data. And so some of the factors they're considering, again, I think I mentioned most of these: sample preparation methods and the sites where they were prepared. How much sequencing do you really need? This is a good question. Generally you want more sequencing depth, but at some point you can go too far and you have so much data that it confuses the algorithms, and that's bad too. So how much data, when is more data better, and when is it not? How much, there's different measures for pre-processing the data before you actually get into the analysis, and how do those affect the results? And then there's potentially many others that they could try. This is an ongoing study. At this point, they've looked at sort of 2 of these aspects. One is comparing the outcomes of the transcriptome assembly and analysis across 6 different sites that prepared the sequencing data, and then looking at the effect of the library preparation on the results. So this is what they wanted to do. Their workflow was sort of summarized here, and then I'll show you the detailed one, which you can't, there's too much there to actually read. So this is the high-level view, where you're pre-processing, removing repetitive sequences, things you don't want to look at. You assemble all the transcripts so that you can create, hopefully, ideally entire transcripts, entire genes that were expressed. This is commonly done with a tool called Trinity. And then doing the post-processing quality assessment analysis of the data, and looking at various statistical measures to get an idea of how well you perform. And so this group, the data sets that they wanted to do were some of the largest that had ever been done in terms of transcriptome assemblies, and they wanted to look at the effective depth of sequencing, and so they really wanted to have a lot. Some of these were extremely large data sets in terms of in the context of transcriptome assembly, and which means maybe a terabyte of data per assembly. But there's a lot of different assemblies happening here. So and it's a very long, many weeks. Well, on other systems previously it was many weeks. On bridges it was actually quite a bit faster, even for some of these big data sets where you get it down to a week or so. but anyway, each one of these was a massive undertaking, and so connecting all these pieces into this pipeline and getting the data through them was just really more than this group could wrap their head around. And so Raminder was the one who took this over and helped get this done. Now, we were able to build on some previous work with Trinity Assembly on our other systems, where we laid out sort of the best way to run these on a big memory system, either using RAM disk or on bridges. We can also use local disk that's there. For certain parts of the calculation that really benefit from that, at some points Trinity creates hundreds of thousands and sometimes millions of little files, and so this is really something you don't want to do on a shared file system. And so that part of it we were able to build on for the assembly part, but this is the entire pipeline. And so this just gives you this is just to give you a bird's eye view, or just a sense of the complexity of this, all the different tools involved. There's different memory requirements, there's different ideal numbers of processors for each one of these steps, and the amount of time it takes varies quite a bit across these. And so this was what they wanted to do this project, but they really needed someone with some more computational savvy and familiar with the system to do this. And so Raminder was able to wrangle this through, consulting with me, and running into various issues. Again, the heterogeneity of the tools and their requirements. This was something he initially started to do with workflow tools, with like a gateway tool, thinking towards developing this into a gateway. But he found it quickly it was just too complex to start there, and he just needed to start with the command line and work through with the traditional method. And that was due to that sort of the heterogeneity, but also a lot of these issues with long-running processes taking days to weeks, having to take snapshots, ensure against failure, be able to recover at some point because you don't want to lose weeks of work, and then just moving data. Some of these require local disks, some of them can be run from the shared disk, and then so moving from the scratch shared file system to local disk, then to long-term storage. Just these details that are okay with one or two applications, but when you have 30 of them lined up, you get some bad data somewhere, and that propagates through. Having to verify correctness of data each of these steps is an important part of the process. So he worked through this, and just quickly here, as I'm basically out of time, we have these are some of the initial results they saw as they look across the bottom here: 1, 2, 3, 4, 5, 6. These are the different sites that did the preparation of the data, and these are the results. And so the higher here is better. This is the N50 measure that I talked about at the beginning. So here I am using it. So more contiguous is better. So the higher this number, the better. And this, if you look at this top line, you see that a lot of these sites are kind of similar, but then you have this big dip here at site number 3, and so they're seeing if something happened here at the site number 3, and if you look at the library preparation across here's sites 2, 3, and 5. Again, 3, using their libraries, you get this bad result. But if site 3 used a library from the vendor, then they did fine. So this is sort of pinning down that something in the library preparation for site number 3 is off, and so they can send feedback to that side and determine what's going wrong there. There's lots of other aspects of this that they're still looking at, and they have some preliminary results, but not quite ready to share on the sequencing depths and pre-processing and such. And then they still want to make this transcriptome analysis pipeline available through some sort of gateway, so others don't have to go through the pain that they went through. If you can get it sort of automated and in something like Galaxy, where others just run it. And so basically I'll just have time just to briefly mention that we've been working with Galaxy to enable these types of large shared memory jobs like the Trinity assemblies on their main Galaxy site that has 90,000 users. And so this is a big win. This opens up a lot more people to be able to use these tools where they might not have the computational background to do so. And so they've been running jobs on Stampede for over a year, these smaller jobs, but we just recently created an optimized Trinity Galaxy tool that runs on bridges, which is now active, and this is a snapshot of the screenshot of the Galaxy main site with a Trinity job ready to go there. So that's one step in that pipeline, but there's sort of a complex process. So here's the a lot of complex work that still needs to be done. And then I'll just leave you with just with this a pointer to this other project that's really interesting, where they're looking at sort of doing a critical assessment of all these different tools. One of these main questions was, what tool should I use, and how do I know which one's better? Often the papers that are published, the authors will intensely optimize their own tool and then run the other tools with default settings. And, and of course, they're publishing a paper because their tool obviously is better. And if it wasn't, they'd have nothing to publish. So anyway, there's a lot of this that happens. And so they're looking at, more critically assessing and having a framework for assessing the quality of these tools. So this is a project I was able to work with through this novel of projects of ECSS. It's actually a foreign group based in Germany. They have U.S. collaborators, and they've created a Docker framework where they have various assessment tools that people can run their tools through these containers, to reproducibly look at, where is my tool now in terms of a bunch of different measures. And so here's a bunch of different measures of quality of assembly and that sort of thing that people have submitted for the analysis, and they've sort of turned it into a competition and looking at which tools are performing best, which are the state of the art. So putting something like this into like a Galaxy framework, or sort of like with containers and that sort of thing, is sort of like a pie in the sky eventual idea that we might be able to support on ECSS to help facilitate some of this, both for people running analyses and also for evaluating the quality of their analyses and the tools that they're using. So I'm going to stop there, and if there's a brief question or two, I guess we could take that.
Okay, thank you very much, Phil. Terrific talk. Please feel free to ask Phil 2 or 3 questions.
Boys in the background. So Phil, one of the things that comes to mind, this is Jeff, is with all of the tools out there, it's very confusing to these people that are relatively new to large-scale compute environments and all of the tools of the trade. Some of the bioinformatics community has been putting out small data sets and little tutorials to get people's feet wet. Is that something that you think ECSS should be doing as well? Have like an example data set that's stored on bridges with like a best case example for PacBio data and a best case example for limited data, or how should we be ramping people up to do this?
Yeah, I think that's. I think that is great. I mean, you can on one side you could have some sort of tutorial on the web and point people how to do that, and I think that's definitely a good way to go. Also, encapsulating these things in something like Galaxy, where you sort of have the knowledge built in, I think is another good way to go for people who just want to sort of go with a sort of a standard approach, or an approach that's been shown to be good for particular type of analysis or something. So yeah, I think we could do a lot more. There's certainly a lot more work that could be done to help ease folks into being able to use the resource and be able to have an idea of where to start with with some of these projects. For now, they in a lot of cases they have to come talk with someone like you or me, and we help direct them. But we could do more to help them get started on their own.
Sounds good. Thanks.
Okay, thanks again, Phil. Let's go to Darren. Darren, I made you co-host, so I think you should be able to share your material now.
Okay, how's that look?
Okay, very good. Take it away, Darren.
I'll jump right in here. So I'm going to be talking today about a different style project from what we previously heard. So this is it's a single application a research group has been working with for a number of years. And they were using this to do basic fluid dynamics research. The thrust of the ECSS work is to help this group study these large-scale computational fluid dynamics problems. So one of the basic problems, test cases here is just a basic turbulence in a box. There's still fundamental questions about these kinds of studies, such as the universality of various statistics, and how those universal traits scale with a larger Reynolds number. So these are things that they're trying to study. They're also looking at turbulent boundary layer problems. So these are things that come into play with aerodynamics and various other kinds of simulations. They're looking at droplet laden flows. And so these would be things that involve sprays, that they can involve combustion. So they're trying to study the interactions of small droplets in a turbulent flow. And they're looking at trying to do this for very high Reynolds numbers. So they're looking at petascale type of supercomputers and beyond. So the goal of the collaboration here was to adapt their simulation code, which was initially a 1D domain decomposition code, which I'll get into a little bit later. They could run this thing on approximately 1,000 processors or so, and they wanted to be able to enable much larger scale runs, and therefore much larger grids, giving them higher Reynolds number capabilities and so forth. And so they wanted to try to keep their existing numerical approach, but move to this 2D domain decomposition. And this presented some numerical method problems that we had to resolve. But the advantage of keeping the approach is that we have all these use cases already established. That had fairly good performance. So we had some confidence that if we could do this, that the code would still perform well. They had the boundary conditions that they wanted were supported by this method. So that's another complication that we wouldn't have to deal with if we could do this. And we also had this direct comparison of results that we could do between the working code and with the 1D decomposition and the code that we were trying to implement with the 2D decomposition. So this, I'll kind of briefly talk about the numerical method a little bit. Basically, this is solving the Poisson equation, which is something that's necessary. It's a necessary step in most direct numerical simulation codes. And it's typically a large bottleneck. In a lot of codes, it's addressed using a 3D FFT, and this method is a little bit different. So I'll talk a little bit about the numerical method and then details about the implementation, which might be a little more relevant for...
Hello? 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 Is this Thomas?
Please unmute.
Could,
Yeah, hi, who is this?
Please mute yourself.
could you mute the phone call, possibly?
Thomas, mute yourself, please.
Okay. Okay.
Hey,
Yeah.
Sergio, you can actually mute them if you go to participants and click on
Yeah,
the
I
microphone.
can find him.
Just to consider the paper that I sent you first, the 6 pages.
Okay, well, I'll keep going.
Because what I want
So this is the general equation. It looks nice and neat. Partial differential equation, the Poisson equation. Actually, this is a more general form with this. If the lambda term here is 0, it becomes the Poisson equation, which is typically what we are actually solving in the code. This is the discrete version. So this is what happens when you put it on a mesh, when you put it on a grid. And this is using a typical method that's second order accurate called the central finite difference. And then these indices indicate the ij, k Cartesian grid coordinates. And I won't talk about the boundary conditions. We're using Fourier transforms, so FFTs. In this case, we're not doing a full 3-dimensional FFT, and I'll get into a little bit of why that's important later on. We're doing a 2-dimensional FFT. And this is actually a feature of the original code. So the original 1D code did a 2-dimensional FFT that was parallel. And so we already had that mechanism in place. And so what we do if you were to work this out, write down the central finite difference version of the formula and use FFTs, you're able to construct a linear system, this top line. So that's what's left to solve after you do it. So you do a 2D FFT, and then you have to solve this linear system. And this is what it looks like in matrix. And so this is the numerical approach that we started with. The linear system was solved serially, and the two-dimensional FFT was done in parallel. And so this is basically how the method goes. We do the 2D FFT. Again, that's done in parallel. It involves, you're doing a real to complex. So you end up in complex space with after the FFT is performed, and then you're solving this complex tri-diagonal linear system I showed in the matrix form the slide before. And then you have to go back to real space with the inverse FFT. So that's the basic structure of the method. Now, what we started with was this one-dimensional decomposition. In the case of this code, we were splitting along the y dimension. Just a choice of Cartesian coordinates. It could be any dimension. And the scaling was really efficient. The complexity was low, and with only one parallel dimension, and the code typically scaled very well. The problem is here, though, that the scaling is limited because, for instance, if you're solving a 1024 cubed mesh, you can only run on 1,000 processors maximum in the y direction, because that's just as many grid points as that you have available to split among processors. So even for if you're going to move to a larger mesh, you still can't get enough computational power involved to get a petascale speeds with this approach. The parallel two-dimensional FFT, which is a key component of the solver, I just kind of use the 1D decomposition to illustrate how that works. Is you do the serial FFTs for every J and K index, and then you have to do a global transpose, and that typically involves MPI all to all, which shifts the data layout. You see with the animation, and then once you have that, the global dimension has shifted, then you can do the FFTs along the y dimension. So you start off with the x and z dimensions global, so you can do a FFT along x, and then you transpose so that you can then do the FFTs in the y direction. And it's this transpose operation that's typically the big bottleneck, because that involves MPI all to all. And when you're running on thousands of processors, this starts to dominate the runtime. So I have an illustration here of a two-dimensional domain decomposition layout. Here the y and z dimensions are parallel. Each of these rectangles would represent a different MPI rank. The advantage here is that it gets us to petascale. We're practically unlimited in our scaling. I say practically because the largest meshes that we're looking at doing here are 1,000 cubed, and so even at that level, you're already talking like 60 million possible processors. So practically speaking, we're only running on hundreds of thousands at this time. So this gives us that capability. However, that involves more complexity. We have to come up with another way to parallelize this algorithm. And that's what a lot of the ECSS support that I provided addressed. Now, a completely different method, which we actually looked at, and there's a library that at San Diego was developed called P3D FFT, which also sort of came out of the ECSS support from TeraGrid. This is the common approach to get at these kinds of problems. And if you're going to do all three dimensions, you have to end up doing 4 global transpose operations to go both ways. So 2 of those transposes that I illustrated before for both of the forward and backward FFTs. So what we decided to do for this code in order to preserve the numerical method was to keep the 2D FFT and then try to parallelize the linear solver. That allowed us to reuse code from the 2D FFT that we had already implemented. And the transpose, then we only require the one transpose that I just kind of showed there with the animation. And we don't need to transpose it to make it global in the Z direction. We can keep that dimension parallel and then solve our linear equation. Some background on the numerical method for the parallel linear solver. I won't go into a lot of detail here, but I'll just sort of point out what's important. The way this algorithm works is that you're able to do a parallel problem here where you solve these local solutions. And so these equations, the feature that's important here is that each of these is operating on its own subdomain. And so the equations on the top can be done in parallel on each of these Z subdomains. So these are very fast. And the only communication that's required to solve these is nearest neighbor. So this all scales very well. And then the problem is that as you work out what's required to construct your final solution is that you need to, in order to correct for the boundary conditions that you imposed, so you impose these artificial conditions in order to solve these local problems. And so in order to correct for that, you have to solve this smaller linear system to apply these global corrections. And so this is really the bottleneck of this parallelization technique. In this reduced linear system that I have on this slide is actually in the same form as the linear system that we solved serially previously for the original method. So it has the same linear form, which is good because then we can reuse code again. We can reuse the serial solver, but we're solving a much smaller linear problem. And this is just to apply these global corrections. So these are the kinds of things we were dealing with, is how do we implement this at scale? Is it going to scale? Is this going to kill the whole approach and then we just have to scrap this and use something else? And these were things that the research team at University of Washington just they weren't equipped to be able to implement this at the kind of scale that they were trying to deal with. And so I was able to implement this at NCSA and test it and give them feedback on whether or not this was a good approach. And so the overall approach went as follows. So we're trying to implement this parallel linear solver, and so we're trying to do it efficiently. So we initialize as many things as we can ahead of time. And then we compute the localized solutions. That all happens very fast and very good scaling. Nearest neighbor communication is required, but that scales well at least. And then we do the global correction, which does not scale well. But we can reuse the same linear solver that they had already had in place with the original code, so that's nice. And then we put it all together for the final solution, and again, that's a parallel, good scaling, completely parallel with very minimal nearest neighbor communication. So just an overview of the solver. We do our FFT operations. We do a global transpose in step 2. We do the second FFT. We execute the parallel linear solver. And then we do the inverse FFT along the global dimension. We do the second transpose to get back. And then we finally get back into physical space by doing the final FFT operation. And then they have their physical solution to the Poisson equation, which they go on to using the Navier-Stokes. Now here's a little bit of performance data from Stampede. And this shows the breakdown of the contribution of the two components. So here the red curve is the overall wall clock time for the Poisson solver. So this is the solver that's at the core of the Navier-Stokes solution. And I broke it down into the FFT component and the linear solver component. So you see here that the FFT still dominates the solution time. And we also see, so this was all of these data points were collected running on approximately 16,000 ranks on Stampede. The mesh size was 4,096 cubed. And what this is showing here is that each of these points represents a distinct run. So the point on the left, where the K ranks is equal to 4, would be, that means that there would be 4 ranks along the K dimension, which is the dimension of the parallel linear solver. And then there would be 4,096 ranks along the Y dimension, so along the FFT, the parallel FFT dimension. And then you can run this, you can change that configuration. So part of the challenge of running these kinds of codes is that you want to try to find the optimal configuration. And so we tried several different ones for this mesh. And this shows varying the configuration all the way to where we have more ranks in the K dimension. So we have 512 ranks in the K dimension on the far right. And so what you see is that for the points on the far right, that's when the linear solver starts to break down. We know it doesn't scale great, but the point is that we can get enough scaling out of it to be able to run on 16,000 processors in this case, which wasn't even possible before. We could only run on 4,000 for this mesh size. But we're seeing that as we push the envelope on the number of processors up for this parallel linear solver, that it starts to dominate the runtime and eclipse the FFT time. And so that's what the challenge is going forward, is how to suppress that tendency. And so the lessons learned on this were that, first of all, we had to take this approach of using bulk operations on the entire set of vectors that we need to solve. So, we applied a numerical method that was developed to solve one vector, but we have a whole slab of vectors that we need to solve. So we use bulk MPI operations to do the communication. And then another neat trick that we developed was the ability to do multi-level parallel decomposition, is what I call distributed gathering. So what that entails is that when we do this global correction, that we don't just gather all the solutions to one processor. What we gather, we parallelize the index space, and we gather subsets of the vectors so that we can still use all of the processors at the same time. And I have a little animation I hope might illustrate that a little better than me explaining it. And finally, another significant performance boost was gained by using non-blocking collectives that was introduced in the MPI-3 standard. In this case, the MPI gather and scatter non-blocking collectives. And so that was a significant boost to performance as well. So I'll show these. So on the left here, I have an illustration of a root gather operation. I'll try to. Now, what the animation is illustrating here is that the Z dimension. What's happening here is that each distinct Z dimension is being like these coefficients that we need to solve for are being gathered to a root. So So we have, in this case, 4 root, so k equals 0 processors that are gathering all the vectors that need to be solved. And so when that happens, once that gather is complete, then you have to go through the full index space of x and y, solve all of those linear systems, and then push them back out in the opposite of that way. Now this distributed gather works a little differently. What happens is you impose you reparallelize the y index space. And so you say for this set of k processors, only gather some of the vectors, and then gather the rest over on this one, and so on, so that once that gather operation is completed, you can simultaneously solve those systems and then send them back out. This way you have all the ranks involved, at the same time being able to solve these linear systems instead of just these 4 over here. And for the non-blocking communication, this is just a simple illustration, a little snippet of code that shows that the way we implement the gathers with a simple loop where, since this is a non-blocking call, this loop can post all of these gathers simultaneously, whereas before MPI 3 there was no collective non-blocking collective, so you'd have to sequentially do these gathers, and since these operations are happening on distinct processor groups, we're able to put all the data out at the same time and get the overall bandwidth to increase. And this is another snippet of code from Stampede, and this shows the effect of the MPI non-blocking. We see this yellow line would be the root gather method that I showed that was on the left side of the animation. The blue line is the distributed gather, and then the magenta line here shows the same distributed gather technique but using the non-blocking collectives. And so we were able to get access to Blue Waters and run some scaling tests on a larger mesh that we couldn't run on Stampede. 8,192 cubed. We started by implementing the 3D-FFT approach that I mentioned before, P3D-FFT, to solve the Poisson equation as a baseline performance. And the first set of performance numbers we got was from the implementation that did the simpler root gather approach. And we saw that we were getting similar performance from the 3D-FFT. But once we switched to the distributed gather with the non-blocking collectives, we were able to boost the performance significantly over the 3D-FFT. And this final performance curve here is just to give us an idea of how close we are to sort of optimal performance, which would involve none of this costly global correction. We just skipped that. So the blue curve is actually not realistic, but it's an illustration of like the scaling floor. I think I'm getting a little short on time, so I'll go ahead and stop with this slide. And if there's any questions.
Thank you very much, Darren. That was, I think, a great talk. We have time for 2 questions or 3 for Darren.
I have a question. This is Yang Wang from PSC. Do you have any write-up of this approach?
Yeah, so we have, there were 2 conference papers that we submitted. I don't know if they're actually available, but I can get you a copy of those.
Yeah, would you please? Yes, I'm quite interested in this.
Yeah, it doesn't have all the numerical detail. We're actually working on a journal computational physics manuscript that described this more fully. But it has a lot of detail, and I can answer any questions that you have as far as that goes. So yeah, I can post a link to the to the conference papers if you want to maybe send me an email.
Maybe you could just send it to, I don't know, ECSS all, you know, if you have the link, the URL.
Yeah. I can try that, yeah.
Okay, thank you, thank you.
Darren, I have a very non-technical question. I was wondering how you created that nice animation about how the matrix was broken up.
Yeah, that was, I actually used Mathematica for that. And it was very painful. I just kind of was painstakingly came up with these clunky formulas that created that, and then I just pieced them together in an animation.
And you did that just for this presentation?
I didn't. I actually did that for the presentation of a conference paper in Montreal that I did in August.
Okay.
So I recycled some slides.
Okay, maybe one more question if we have any. If not, then thank you very much to, I think we had 2 outstanding talks today. And we'll see you again next month, I guess, unless it's preempted by supercomputing. I'll let Nancy deal with that. Thank you very much. Bye-bye.
Thank you, bye-bye.
Thanks, Sergio.
Thank you. Bye.
Bye-bye. Thank you. Thank you, everyone. Bye-bye.